Okay, we have some more already. Maybe we just start with the introduction and then slowly I think the people will join if you agree with that.
Sure, sure.
Sure. Okay, perfect. So good afternoon. My name is Diego Flores from BMK. I will moderate this webinar and I am happy to welcome each of you. This session will last about 30 minutes with 20 minutes dedicated to a presentation and 10 minutes dedicated for your questions. Feel free to submit your inquiries using the Q&A button at the bottom of your screen. If we don't answer your questions during the session, we will email you a detailed response. Additionally, we will send out the slides and the webinar recording afterward. So before we dive in, let me introduce our distinguished speaker for today, Dr. Sara Bass, a territory manager at BMK. Sara has a PhD in molecular and cell biology. She has expertise in transcriptomics and epigenomics for chemotherapeutic research, and she has extensive experience in genomics project management from initial design to delivery and ongoing support. With that being said, it is my pleasure to hand things over to her. Sara, please, the stage is yours.
Hi, everyone. Thank you, Diogo, for the introduction, and thank you all for joining today's webinar. I'm excited to discuss one of the most commonly asked questions about mRNA-seq, which is how much RNA do you really need to get high-quality data? This topic is crucial for anyone planning RNA-seq experiments and looking to balance sample availability that sometimes is not easy, and we know about it, with sequencing depth and data quality. This is part of our 15 minutes quick and casual webinars. So the plan here is not to spend 1 hour extensively talking about the biochemistry of the sequencing reaction, but it's rather going to very specific key points that we often find that are pain points for researchers when they are planning their mRNA-seq experiments. And for example, these points could be like, how much data do I need? Or what if I don't have enough RNA? So let's jump right into it. First, what is RNA-seq, especially mRNA-seq? This seems like a basic question, but it's often always worth clarifying. mRNA-seq refers to a technique that provides a snapshot of the actively transcribed genes in a cell or tissue at a given time. This is not to be confused with the study of all RNA molecules in the cell that sometimes is called the whole transcriptome. Here you have actually a pie chart of the different types of RNAs in a mammalian cell, and you can appreciate that mRNA is not a big percentage. However, it's this percentage that has the actively transcribed genes, and assessing those genes is one of the major goals for transcriptomic researchers. You can do many things with your mRNA-seq data, but the two major analyses are the differential expression gene analysis, so telling which genes are upregulated or downregulated between two given samples or between two groups of samples, and the functional annotation of those differently expressed genes. And for example, if the difference between your control samples and your treatment samples, if you have an upregulation of genes related to cell division, or perhaps a downregulation of genes related to cell adhesion pathways. If you are interested on the bioinformatics part of the mRNA-seq, we have some other recorded webinars in our website, and you can just go and watch them. But now, how can you technically get mRNA-seq data? Nowadays, there are two major strategies. So you have the poly-A-based capture and the ribosomal RNA depletion strategy. The poly-A-base captured uses poly-T bits that catch the poly-A tail of mRNAs. This makes it highly specific, and it's currently the most cost-effective method for mRNA-seq. However, as it has some points that might be considered some cons to some researchers, and this is like being limited to poly-A mRNAs, so it's not suitable for prokaryotic RNAs, and it's very dependent on the RNA quality, and this is if the poly-A tail is very degraded, the mRNA may not be captured. The ribosomal RNA depletion method works in a different way. Basically, you deplete the ribosomal RNA from your total RNA, and thus you end up with an RNA that's enriched for mRNA, long non-coding RNA, and circRNA, which then we process to sequence everything. This method allows for the capture of non-polyadenylated RNA. It works a bit better when we are talking about degraded RNA, and the most important, it provides you with broader knowledge of the transcriptome coverage. Nonetheless, it has higher cost, it has the potential for higher background data, and you may want to clean this during the data analysis, and it's less selective for mRNA, which of course can be either a pro or a con, depending on your research goals. In this webinar, we'll focus mostly on the poly-A-based capture. And the general principle is the following. So we have the RNA, we capture the mRNA with poly-T bits, we fragment it, we perform the first and the second cDNA amplifications. The ends are repaired so that the sequencing adapters can ligate. The cDNA is purified and the final library is amplified. We perform the quality control before we start the sequence, just to make sure that everything is okay. And if all is well, we perform the sequencing. In this standard RNA sequencing, you will not get strand-specific data. You will have both data of both of the strands, which on the case of the mRNA, it does not provide a huge advantage. So of course, it provides advantage because the annotation will be much better, but it's not like on the case of long non-coding RNAs, in which you definitely need the strand direction so that you know the direction of the long non-coding. What we get also frequently asked is how much data is needed for mRNA-seq experiments. And thus, I wanted to quickly run you through the rationale. So the more you sequence, the more data you get, the more line reads you have, and this leads to more robust statistic data analysis. And this means, of course, increased accuracy and biological significance in gene expression quantification. Usually, 20 million parent reads per sample provides a good coverage for most expressed genes. But if you're interested in low abundant transcripts, you may want to opt for 40, 50 million parent reads per sample. Nonetheless, this is the general rule of thumb. However, we highly recommend to always check the literature of your field to see what other peers have done and to see what different types of scientific journals normally accept for publication. What are the factors that are influencing more the RNA input requirements? And this is a very, very, very broad question. The easiest way to reply to this is basically it will depend on the sensitivity of the sequencing platform. This is both the sequencer itself and the library preparation kits that we use. And nowadays there is a big variety here, and we will address also this further ahead in the presentation. If you want short reads or long reads. This is also very important because in the second case, you'll need much more RNA. And of course, the type of capture. Secondly, the quality of the RNA itself. Because high quality RNA is essential for good mRNA-seq experiments, but sometimes you have a lower quality RNA, so you might want to send a little bit more RNA to help you get more data. But of course, in the end, degradation and possible fragmentation of the RNA may impact read length and data interpretation, even if you send more RNA. But now talking about the actual numbers. So different platforms and different kits require different input amounts, as I just mentioned, but these are the ones that we recommend at BMKG as a standard. Of course, if you can send more, the better. If you cannot meet those requirements, but you are very close, we recommend you reaching out to your regional account manager to discuss if we can still process your RNA as a standard. I have copied here also some best practices to have a good RNA, and they may seem basic, especially for researchers that already do transcriptomics for quite some years now, but it's always good to remember. So always have or always try to aim to have a good tissue or cells starting amount, because no matter if you're using the best kit on the market, if you're not starting with a good amount of RNA, it's virtually impossible to get a good amount of cDNA at the end of the process. Use fresh or properly stored tissues. Also avoid freeze-draw cycles. Use optimized Trizol or column-based kits for your specific samples. Always assess RNA quality and concentration. And for concentration, qubit is ideal. Maintain an RNase-free conditions throughout the process. Work quickly on ice. Use DNase treatment if necessary. And the specific point here that I would like to mention is that many kits do not have the DNase inside the kit, so you have to buy it as a separate reagent, even though they have the DNase protocol inside the kit itself. And then always store RNA at -80. Then I also wanted to mention the specific case of blood mRNA-seq. It's something very specific that will, of course, not be relevant to all the researchers, but in blood RNA-seq, you have lots of molecules from globulin mRNA that can account to up of 70% of the total RNA. So it's highly recommended to do globulin mRNA depletion kits. Otherwise, you'll look at your final data and you'll find that almost all of the reads are from globulin. If you don't have these kits in your lab, there is no problem. We can also do them at BMKG. You just have to let us know in advance. And now I think the question that everyone was thinking when they joined this webinar, the $1 million question. So what if I don't have enough RNA? And unfortunately, this is the case of some researchers due to the sample itself. For example, exosomes, FFPE, small target tissues, and the list goes on. There is a solution for this problem, like low input and ultra low input kits. But before I talk about them, I want to talk about the potential challenge you may face when using them. Because as there is a very low amount of RNA molecules in the sample, so in the tube, in the Eppendorf tube that you send us, there is increased amplification bias. There will be reduced complexity of the library. So this means that you have few unique reads, transcripts detected. Overall, it might happen to have low quality reads, leading to challenges in data interpretation. And additionally, as we are talking about such little RNA, the kits are not 100% efficient, so the library preparation might simply fail sometimes. It's not common because these kits were designed for low input samples, but this is always something to have in mind when planning your experiments. And these are the numbers that we recommend at BMK for low input, ultra low input, and exosomal low input. The strategy is always to try to do the kit that best fits your requirements. So, for example, if you have a sample with 100 nanograms of total RNA, it is recommended that you do the low input, that it has a minimum of 30 nanograms, even though 100 nanograms also fits within the ultra low input requirements. If you have any question about this, or if you want to discuss it, we of course are always open to it. We can book specific and one-to-one meetings with researchers to try to see which is the best kit for them, for their specific project. So you just have to ask your regional account manager and he will do this assessment with you. Before ending this webinar, I wanted to give you a brief overview of the major differences in the protocols of these kits. So, the low input kit uses a strand-specific approach with high efficiency in the adapter ligation and the bit step. So basically, these two reagents are slightly different than the standard one, and this enables a lower input requirements. Then the ultra low input and the exosomal already show slightly more differences. So the ultra low input, instead of using poly-T bits, they use oligo-DT primers, as these have been shown to increase efficiency versus comparing to normal T beads reagents. This kit also has a direct amplification of the second strand of the cDNA with TSO primer. This means that it does template switching, and this enhances the efficiency over traditional random priming. The exosomal kit also do the template switching of the second cDNA strand, but the core difference here is that it does not do poly-A-based capture. It does a depletion of the ribosomal RNA because in the case of exosomals, as exosomes do not have lots of ribosomal RNA, we can remove it much more easily. So it's suitable for these types of low input projects. And in the end, you will end up with data of both mRNA, long non-coding, and circRNA. Therefore, here are the key takeaways for this webinar. So first of all, plan ahead based on your research. So how much depth, sensitivity, coverage you need. Always aim to have the best and high quality, quality and quantity of RNA. Use appropriate services for low input, but please be aware of the trade-offs. And always quantify and assess RNA quality before proceeding with the sequencing, just to avoid wasting time and resources. As we wrap up, I'd like to give you a very quick overview of our full range of services. So from short-read sequencing services with Illumina to single-cell and spatial transcriptomes, we are here to support every step of your research expertise and innovation. We're working on whether you're working on genomics, transcriptomics, microbiomics, epigenomics, or tailored solutions and research center approaches ensure high quality results. Thank you for joining today's session. I hope this has helped you plan your future mRNA-seq experiments. We'll now open the Q&A section. If you have any further questions or need advice on your specific projects, you can just write here on Zoom, or you can also send an email either to info@bmkgene.com or directly to me at sara.fash@bmkgene.com. Thank you.
Perfect. Thank you. Thank you, Sarah, for such a fantastic presentation. And we do receive some questions that we are now planning to answer. So the first question is, can I use the low input kit for prokaryotic mRNA sequencing?
For prokaryotic mRNA sequencing.
Yes.
The kits that are based on low input and poly-A capture, no, because the prokaryotic cells do not have, most of the RNAs do not have poly-A tail in prokaryotic mRNA. Then you have the case of the kits that use low input with ribosomal depletion. In principle, you might think that it will be possible, but no, because the depletion here in these exosomal low input kits is very mild, because normally exosomes don't have much ribosomal RNA to deplete, and so it's not efficient in depleting ribosomal RNA from bacteria. So unfortunately, no.
Perfect. The second question is, why is the RIN value of exosomal RNA low?
This is, yes, I'm in the perfect slide for that. It's because I think I've mentioned it one time in the presentation. So exosomal RNA don't have much ribosomes there, so we cannot do a proper RNA quantification, because RNA quantification is based in several factors, including ribosomal RNA. So if you don't have enough and good quantities of ribosomal RNA, you cannot get good RIN numbers. So it's normal to have this type of values in these types of service. If you have a high RIN value in exosomal services, you might want to check your protocol in case there is a contamination with whole cell ribosomal RNA.
Thank you very much. Another question, can I decide the service after you do the QC at your site?
Yes and no. So it depends on this service. So if we are talking about the first one, the standard and the low input, yes, there is no problem. If we are talking about the ultra low and the exosomal, as we are speaking of such little RNA, we normally don't do the quality control before the library. So what we do is that we get the RNA, we put everything to library preparation and amplification, and then we give you a quality control after the library. So if it went okay, you can advance with the sequencing. If not, we do not recommend. So, but after you have done this, in the case, for instance, of the ultra low input, you cannot go back for the standard one.
Okay. Another question, can we send samples without the use of DNA sequencing?
Yes, but yes, it's possible. But again, this might take into consideration the values of the quality and the integrity, because DNA will will cause some alteration in these 260, 280, and 260, 230 values. So they will not have perfect ratios. What does this mean in the end? In the end is that when we are amplifying the library, even though we do poly-A selection. We will enrich for the RNA, but there might be some contamination of the molecules of DNA. That's why we recommend doing it before sending the samples. However, technically, we can do it, but in the end, you may want to just double check the data to confirm that it does not have much reads of the genome.
Perfect, thank you. So if we don't answer all the questions, we will email you with a detailed response. And as I said before, additionally, we will send out the slides and the webinar recording afterward. So as we finish today's session, we are happy for your participation. Please look out for the follow-up email with a webinar recording and summary, and make sure to check out our social media page for future webinars. Thank you again and have a nice day.
Thank you. Thank you, everyone.
Thank you.